for this winter. And in this webinar, I hope to show you how our software tools can help you make the leap from your drug targets to lead molecules, demonstrating the computational solutions we have in place to accelerate your translational research. So in general guidelines, behind the scenes, we'll be muting your speakers. That's just to keep things not too troublesome. If you do have a question, please feel free to type it into the chat box. Marcus is there to answer any queries that you might have as you go along. We'll be recording the session and be providing that to you for your own personal review, should you wish to do so. So today I'm going to run through a very brief introduction and then move on to describe in more detail a couple of application scenarios where the software that we offer, and perhaps more importantly for the job that I do, the service that I can provide, will be helpful to you and your organizations. At the end of each session, I'll pause briefly, obviously depending on time constraints, to answer any questions that you might have. But please do feel free to use the chat box as we go along. So, first off, and just to introduce myself, my name's Alistair. I'm a medicinal chemist. Spent my career, started in academia and worked with a couple of small UK-based organisations, and have been working with Biosolve IT for over 6 months now. In my role, I provide our clients with expert comp chem support to complement their in-house expertise and bring the software solutions that we provide to bear on their problems. Our tools are widely used in the pharma industry. This is just an outline of all the tools we provide. So our main package is Lead IT. Hide, we do some fragment-based software solutions as well, and some ligand-based approaches also. We also have interfaces to the more common pipeline tools that you might be aware of, Pipeline Pilot and 9. You can keep up to date with what's happening with software, new releases, updates via our social media channels. You should feel free to join those groups. We don't post very heavily and you won't be spammed. We just make our customers aware of updates, especially some new tools that emerge that are often available for free downloads. So perhaps moving on to the more meat and bones of the talk. If you start thinking about what potential applications, generic scenarios you might find yourself in as a drug discovery scientist, you may well be in a situation where you have no known actives and no target structure. In that kind of situation, what you're probably going to start with is just a physical screening campaign. However, you may find yourself in a situation where you have a 3D structure of your target, but again, no known actives. And in that scenario, what you might envisage doing is some kind of virtual screening exercise. This will form the majority of my talk today. I'll go through some of the virtual screening services and software solutions that we can provide. If you find yourself in the first situation where you have no actives but no target structure, what you can do often is generate a homology model and use that as a platform for virtual screening. We don't have an in-house proprietary homology modeling software package, but we do have access to technology that would facilitate us doing that, and we could then use that as a platform for virtual screening. Alternatively, you might have small molecule hits or known actives, but no target structure. In that scenario, you may well do some similarity searching or scaffold hopping. These are obviously advantageous in other ways. They're generally much quicker than 3D virtual screens. And the fourth situation you might find yourself in is when you do have known actives, a 3D target with the structure of the target, and then you may well just anticipate using all of the above scenarios. So, as I said, I'd be focusing on the situation in the first half of the talk where we have no known actives, but we do have a 3D structure of the target. And I want to outline what we can achieve here, how we go about doing that, and how particularly BioSolve IT can help you in that situation. So perhaps most simplistically, we can take a target and identify druggable sites and assess their potential for druggability. I'll talk a little bit more about that too. We can use online data sources to guide the models that we build. I'll outline some best practices in virtual screening, how you might go about building small molecule databases, especially those with reliable 3D structures, and spend some time talking about how we go about executing a virtual screen. Of course, once you've completed your virtual screen, you might then be interested in expanding the subsequent search space, taking those hits and finding close analogues that you might also consider screening. So, you think you found a great new drug target, and you have a crystal structure, but then what? What I wanted to do was make this a bit more real world. So I decided to build up this pretend case study where this academic group have discovered that this protein SND1 promotes tumor angiogenesis in some hepatocellular carcinoma, and use this as a basis for how we might go about building a project. So the first step that I normally take is to search an online resource, UniProt, for the target of interest, and check that for natural variants of the target, and alternatively we might be interested in relevant mutant forms. What we can also do there is check that the human form of the protein is not different, or if it is different, how different it is from an animal species that might be relevant to subsequent model studies. And we usually achieve that using a BLAST homology search. And the other nice thing about UniProt is that it will have a direct link to any 3D structures if they are available. So I thought I'd just quickly show you that in Firefox here. So here's a browser window. All I do is type in SND1. Obviously I've done this before. And the first hit here is from the mouse. You'll see, I'll just highlight the spotlight here. So the first hit's from a mouse. We're not so interested in that at the moment. The second hit is from an uncharacterized protein. So again, that's probably something that's not going to be interesting given that it is uncharacterized. But the third hit here is our target of interest, the human form of SND1. So if I click on that, what appears is the information about that specific target. So first up, we have some synonyms, excuse me, for that target, and gives us some information about the length of that protein, general function, and what I became interested in here, we've got some information about different domains within that protein. So what jumped out at me was this pseudodomain, because I was working on another project which was also interested in looking at pseudodomains. Further down, what we find is the domain information. So telling us that this pseudodomain, which might be of interest, is from amino acids 729 to 787. And then if we keep on going down, one other thing that jumps out is that we have an amino acid at 781, so within our pseudodomain that can be phosphorylated. That's just something to bear in mind when we're looking at the structure, that we want to make sure that the 3D structure that we're looking at is in the correct phosphorylation state, because quite often that will cause quite a large conformational change in the protein. What we can do within the pseudodomain, if we click on the link here, that will take us directly to a BLAST search, which I have pre-performed. We should have this one here, which tells us which of the proteins across a number of species are most similar to our target of interest. So we can filter those by species, and if we look at mouse, because I'm assuming we're doing an animal model in a mouse, we just have to scroll down and down and down past all the animal until we eventually come to the mouse. So what I would then probably do, if I just switch back to my window here, is just to summarize that, is okay, we found a protein of interest, we've determined if any 3D structure is available, and we found it as a pseudodomain. This is a novel and interesting area for us to target, but there are no known binders at this region. So what I would then do is jump into find out those 3D structures, and those are listed further down on the original page. So if we keep on going down, what we'll find is that at the bottom here we have a link to any 3D structures that are available. So you can see that a number of these encompass those residues from the pseudodomain, and what I would then do is just look through these one by one and find out which was perhaps most interesting or relevant to my needs. So I did that, and we have a very high resolution structure, which is 2HQX, which is resolved 1.42 angstroms, but one drawback with that one is that there's no ligand or substrate present in that structure. However, another form with a peptide, also at very good resolution, is 3OMC, and the fact that we have that peptide substrate in place simplifies the defining of the binding site when we come to build up the definition of that binding site for virtual screening studies. So now we've got a potential PDB file to work with. We might then think about, okay, well, I'm interested in this pseudodomain, but we want to assess, is that a druggable site, and perhaps have some evaluation of that binding site. And for this, there are a couple of tools. One is called DogSite Scorer from the Center for Bioinformatics in Hamburg, who we collaborate with extensively. And all you need to do there is enter a PDB code. There's also one from the European Bioinformatics Institute called Drugability, and they're available at the links there. So what I did was take a look at this protein SND1 within 3OMC. So if we open up DogSite Scorer, you see it's a very simple interface. I just have to input my PDB code here, and I'll just quickly do that, 3OMC. And then it'll ask us which pockets we want to analyze, how detailed we want to be, and then if I click on the whole protein and assess pockets, sub-pockets, as well as druggability, I can then click on calculate and analyze. That takes maybe a couple of minutes, so I pre-ran that search, and this is what you end up with, which is a diagram of the sites that software has identified as being binding pockets, and it's also given each of those a drug-like or a drug score. So because I've been working with this for a while, I know that this region here, P3, is our tutor domain of interest, which scores at 0.55. Obviously not as good as a 0.7 or 0.74, but one thing to bear in mind here is that, okay, this is our original domain. When I look at that, what I see is that this is our tutor domain pocket. We also have an adjacent sub-pocket from what the software has identified as pocket 1 rather than pocket 3. So that's this red blob here, which you can see is directly adjacent. And because we can get a score for the sub-pockets, which I'll just down here, you can see that pocket 1, so if I define sub-pockets of 1, we see in orange here, we get a score of 0.22 for that sub-pocket. So putting those two scores together, 0.55 plus 0.22, we're probably going to be up in the 0.7 region, the green region of druggability, which is very encouraging. I did the same thing with the EBI tool, druggability. It picks out the whole pocket in this instance and then scores that as being a chemically tractable pocket. Okay. So in order to find these structures at the rcsb.org, it's very simple. You just type in their search box, your SND1, and or your code identified from the UniProt here, 3OMC, and then here is a direct link to download those files. So what I've done for you is to generate a 3D model of the whole protein, and you can get a much better okay, sorry about that. Back to so. Apologies for Windows wishing to update, it's just the wrong moment. What you can see is that binding pocket, and it looks very nice. You can imagine small molecules binding there, really being quite a nice little binding pocket. So another way you can visualize the binding site is with our tool pose view. If you pull out a PDB structure which has a bound ligand, there's a link beside the ligand to a direct pose view analysis of that pocket, and this can often give you an insight into specific bonding interactions that you might wish to recapitulate in your docking exercise. So I mentioned earlier about picking the difference between human and mouse forms. What I've done for you here is look at more detail in the blast alignments of the human versus mouse of the tutor domains. And what we can do is line those up, and what we see immediately is that they're very similar overall with some very conservative mutations, only 3 in total. And I picked those out. Here in red, we have the first one, which is isoleucine, which in the mouse is a valine. This second mutation is from a valine to an isoleucine in the mouse. And down here is the serine, which I mentioned earlier as being phosphorylated. And we can see that none of those different amino acids in mouse are involved in the binding site at all. And so we're then going to be fairly confident that if we do an animal study in a mouse, that there should be no trouble with the mouse target being in any way different. So that's really most of the background. If you start thinking about starting your virtual screen, you really need 2 main components. The first thing that we require is some kind of receptor definition, and within our software lead IT, this is in the form of a project file. And the way we go about that is was demonstrated by my colleague Marcus in one of the earlier webinars, but I'll briefly demonstrate that again for you. So what we have here is our lead IT software, and I was going to work through this backwards, but I'll do it forwards. So you can download your target of interest directly from the PDB server within lead IT. I've loaded it from a saved version. It will give you some blurb about the target and essentially just show the text of that PDB file. We then have a choice of chains. You can use them both, you can use one. Here I've chosen the one in blue because when you flick to the next step, that's to choose a reference ligand. And with the blue chain, what we see is that the reference ligand is bigger than in the other chain. And for that reason alone, I picked that specific chain of A or B. So the next step says, okay, we've got a ligand. It's in the binding site. How much space around that ligand do we want to use to define our binding site? The default is 6.5, and then I click next, and it's really very simple. There's a lot of technology gone in behind the scenes here to make sure that when you define the binding site, normally it's an extremely straightforward process. What you can do if you want to ensure that everything is as it should be, you can then dig in, and usually what you want to look for here is that the software has correctly assigned the residues like asparagine, where the crystallography might not be sufficiently well resolved to tell the difference between an oxygen and a nitrogen and amino acids, where those two elements, so you've got asparagine, you've got the carboxamide at the end, it might be very difficult to tell those apart crystallographically, that those are in the right orientations. And so you can go through that and make sure those are all well aligned. You might wish to pick out water molecules from the crystal structure, or include them or not include them within your receptor definition. And here we have the reference ligand. You may wish to include other small molecules as part of your receptor definition. And then I click go. It asks me to save it, and really that is me finished. I now defined the binding pocket, and what this is based on is, of course, a peptide fragment which shows the. If I zoom right in here, you can see it's a dimethylated arginine residue, which is what the tutor domain recognizes. And that really is, in a nutshell, all you really need to do to define the project file. What you can do within that is define pharmacophore features and incorporate them into your docking studies. I'm not going to do that today, but it's really quite simple to set up, and be happy to do that for you, should you wish. So switching back to the main element of my presentation. The next step, so we've defined our binding site. Now all we need is some small molecule libraries to dock in there. So one alternative is zinc, which is a variety of large number of collections, a variety of filtered. You've got drug-like, lead-like, fragment-like. What I tend to use is one called e-molecules. It's about 6 billion molecules, all of which should be commercially available, and they're purchasable directly via e-molecules website, so it really facilitates customer purchasing. It makes things nice and smooth. So once you've decided which collection you're going to use, you download that from e-molecules. But these will be in 2D format, so 2D STFs or SMILES strings. And in order to dock molecules, you need 3D coordinates. So there's a number of tools you can use for this, and we have our own in-house tool which performs that task very well. So now we've got both of the elements we need. what we have to do is have in place a system by which we can automate this process. What I use is something called KNIME, which is very widely used in the cheminformatics community. It's available for free download, it's cross-platform, there's lots of really useful stuff you can do in chemistry virtual screening with KNIME. And it's, of course, somewhat similar to Pilot and Pilot, but has the advantage that it is free for the desktop version. So to summarize the whole process, what we need to do is generate multiple docking poses for each molecule, usually between 3 and 35. What we want to do is then remove the poorly scoring poses, then rescore the good poses, eliminate the duplicate chemical structures, and then rank order the output. So it's very easy to do that within KNIME. Here we read in the molecules. Let's switch point. Never mind. There we go. So here we're using an SDF reader to read in the molecules. We have a node to generate 3D coordinates. This is the project file which I talked about generating within lead IT. This node will then dock. We then filter out the poor poses. We score them with our hide function. We then sort them by how well they score, and then group them by chemical structure. And using a powerful desktop PC, you can screen about 5,000 molecules in exactly this way in about one day. With the speed, really strongly depends on the number of poses you generate per docking per molecule. If you only generate 3 poses here versus 30, this scoring then takes approximately 10 times longer. And if you're going to want to dock and score millions of compounds, you really have to have some form of cluster or parallelized execution behind the scenes there. And that is something that we as an organization will be happy to do for you. Switching back. So once you've done your virtual screening, KNIME also facilitates the post-processing, where you can eliminate molecules with high class scores, with low ligand efficiency, or question popping up there for Freya, but where you can very much look to eliminate false positives. You can use KNIME to look at poses. You can use KNIME to generate reports. You can generate hit clusters, design analogs, virtual chemistry for follow-up studies, and then use KNIME to spit out an SDF or MOL2 file at the end, which can really streamline the purchasing process. And so in terms of the odds of success, we have a glowing testimonial. Our users tell us that we're doing very well. Buyers experience 70% hit rate is likely exceptional. Normally you might expect in the region of 5 to 10% hit rate for your virtual screening. So what I did, just to demonstrate this to you, is to take 1,000 compounds only in the SDF reader and generate 35 docking poses per compound. I had a FlexX scoring filter of better than -10 at this point here, then ran the high scoring and sorted them. Anything better than -35 is usually going to be interesting. So that took about 3 hours, and I've identified 2 hits that score better than -40. So that's very encouraging. And here's one of them here, which is this benzyl substituted oxidizole, which scores -46 kilojoules per mole. The atoms here are coloured by their contribution to the binding affinity. So anything in green is making a positive contribution to binding affinity. Anything in red is making a negative contribution. This automatically suggests to us analogues that we might think about making. So if we screen this compound, turns out to be active, First thing we might do is modify that oxygen atom here into a sulfur. It's more lipophilic, and I did that. That improves the score to minus 49. So that's very encouraging. So I shall perhaps pause there. I'd be happy to answer any questions. If you want to ask a question, then if you could write your highlight that in the chat box, and then Marcus can unmute you, and we can maybe take a couple of questions. Time's moving on.
Okay, this is Marcus speaking. Can you hear me actually loud and clearly?
Sorry, Marcus, could you repeat that? My volume was a bit low.
Oh, okay. Can you hear me? That was essentially the question.
yeah, I can, yeah.
Okay, cool. So let me summarize a little bit. People are asking about what would a good LE be, for example. Maybe you can comment briefly on what LE is at all in principle, and guide them a little bit.
Okay. Ligand efficiency is a measure of affinity per heavy atom in your ligand. And so what the metric is designed to overcome is the bias for molecules to score better simply as a result of their increased size and lipophilicity. So if I quickly switch back to here, ligand efficiency of 0.44 is typically on the good side. Anything better than 0.3 is acceptable. It's a bit complex because the ligand efficiency within NIME is calculated in a slightly different way. But if you're using lead IT, anything better than 0.3, I think, probably gets what a thumb heading towards the up direction. It's generally used in the early stages of discovery to select smaller fragments on the basis of the neatness of fit. And it's, in my experience, quite useful just to keep an eye on the efficiency, that it tracks well with improved potency, and that you're not adding excess lipophilicity without gaining enough potency.
All right. Another question that we had was on your pocket selection, the druggability score, really. Could you perhaps comment on this, how you select, when you know a pocket is really apt for going on with?
So I didn't catch the end of your question there, Marcus. If
So
a pocket is apt for...
The DogSide Scorer website that you showed has a coloring for the druggability of the respective pockets.
Yeah.
And the question is, how do you know what's a good pocket? So is there any threshold for the drug score mentioned? I mean, it runs between 0 and 1. Essentially, 1 is very good and 0 is very bad. Perhaps you can comment on what else a medicinal chemist does when he sees those pockets.
One thing you can do directly within DogSide Scorer is to click on the pocket name, and what that will do is bring up a list of amino acids within that pocket, as well as give you a lipophilic surface score. Generally, if you have more hydrophobic, here we've got hydrophobicity ratio. If you're looking for something that's druggable in general, it's going to be more hydrophobic. You can of course do that for each individual pocket, and it's if you maybe looked at something that was more established as a drug target in terms of the basic drug score, kinase pocket might score at 0.85. So it depends on what kind of target space you're working in, really. If you're working on the edges, the fringes of protein-protein interactions, then 0.55. Other projects we have worked is definitely possible to identify small molecule binders that are very drug-like. And so If you score 0.5, it would be too down on yourself. It's still quite a good score. Okay, well, time's pressing on, and so I should really probably get cracking. The second part of the talk is really focused on ligand-based design, which many companies use to jumpstart research, sometimes called fast follower, or the patent bust, where you take a known binder, often from a competitor, and use that as a basis for a focused search of your biochemical space. Might be because of patentability, ADMET, selectivity, solubility, any of the above. And so what I did here was take an example from my own personal experience, where you might remember from my little CV, I worked at Chromotherapeutics, and this is a project we started, I think, in 2007. We were looking at a double kinase involved in cell cycle, and at that time no Haspin kinase inhibitors were known. And so what we did was undertake a kinase-focused HTS, and that led to the identification of CHR6494. It's a very, very nice molecular weight, it's potent in cells, very attractive hit, except, as is usually the case in kinase space, we've got patentability problems and selectivity concerns. So what I thought I'd just outline is how I would help, or Biosolve IT tools would help in that scenario. So one way we could help is a 2D fuzzy similarity search with feature trees, or F trees. And it's a very useful tool for scaffold hopping because it's very fast, like a two-dimensional search, but you retain the overall topology, connectivity of the individual features of your molecule, but it remains fuzzy. And so it's much more than just a Tanimoto similarity score match. It's got a lot more to it than that. We have F trees available within KNIME. Here's a very simple workflow. I've defined my query molecule. I've drawn that in, loaded that in as a mol2 file. And then what I've done is taken the Chembl database that's available for download from the EBI website, and read that as an SDF. For the sake of simplicity, I took a random selection of 100,000, and I generated the feature trees for those 100,000, and then I computed the similarity, the feature tree similarity between 6494 and then that selection of the Chembl database, and then generated visual mappings of those. So in terms of example feature trees hits, and of course you should remember this is just a very small subsection of potential chemical space. Here's our molecule that we were searching for, and it's picked out this different core. The colors map the atoms that have been mapped as being similar features. And so it's quite obvious that in purple, for example, we have a two-membered ring, five-membered ring with two nitrogens in purple, and for our molecule here we have, again, a five-membered ring with two nitrogens, which has been mapped as being a similar feature. And so you can see that what you start doing, picking out hits which have similar chemical features, but which are maybe connected in slightly different ways, or the nitrogen atoms in the rings might be slightly different. So very useful for scaffold hopping. And this is perhaps one of the best examples here. This hit is taken an indazole ring where we have an H-bond donor and an H-bond acceptor, and it's mapped that as a similar feature to a carboxamide. And in terms of fingerprints, that's a similarity that a fingerprint would likely miss, even though in terms of a kinase inhibitor, you might well see those as a very common hinge binding motif. So that's just to show how feature trees might have helped us take the jump from our lead series. We could have bought or synthesized these and bioassayed them as potential new start points. Another element of the feature trees is that we can create what are known as fragment spaces, and this allows us to undertake the de novo assembly of fragments to generate novel molecules. And these fragments come from combinatorial libraries. We can take compound sets and shred them based on retrosynthetically valid rules, so that when it comes to the forward synthesis, that those are realistic reactions. And we can also shred known actives, so you're going to enrich your fragment collection with elements, fragments that are commonly found against your target. And what we can also do is define the limits of similarity between hits themselves, so we can build more diversity into any chemical space we want to look at. So this can be done, unsurprisingly, in KNIME again. It's very simple. I just read in my fragment space, and I've got a search node as well. And what I've done is defined that I want to find 5 closest hits, which have a similarity to my query molecule of better than 0.9, but which themselves are no more than 0.9 similar to each other. And doing that, I have to apologise for the slightly small size. You can see that we're picking out some really quite structurally diverse compounds here. And if we were looking to do a scaffold hop or morph, that this is generating some really quite distinct chemical motifs than before. And so if we're looking to break out of a chemical local minimum, this might be a really useful tool. What we can also do is in silico chemistry. And so if we take the hit molecule, the chemists among you will see that this is very reasonable. We would take a this halogenated heterocycle, we would do a nucleophilic substitution at this position here, then some kind of palladium catalyzed coupling here, and then we would probably be rapidly accessing a large number of analogues. So we have in-house a huge number of chemical reactions already coded, which we can use in KNIME. And what we can do is choose reliable suppliers of building blocks. We can then filter those molecule sets using our tool Mona and pick out the ones for our particular chemistry of interest. And we can use those to enumerate virtual libraries. And then we can then filter the output set by any molecular properties we might have, or substructure or SAR feature. And the reason that becomes important is that if you take a very simple pair of chemical reactions that I demonstrate here in KNIME, so here is where the first reaction takes place, takes 2 input sets of molecules, and puts them together to generate one product, which is then the input for a second reaction, and then writes out an SDF file. So if you only use Sigma Aldrich, what you can find if you look at their medchem catalogs online, you can have 999 boronic acids or boronates. You can buy, if you only focus on primary aliphatic amines, 972. And so even just picking 2 limited compound sets, you're already generating over 900,000 unique virtual products. And of course, not all of those would be of interest. You might want to filter those by some log P filter, some molecular weight filter, whatever your in-house rules would be. And where it becomes really interesting is that if you have a target structure, at this time we were doing this project, we didn't, but you could then take a subset of those products, doc, score, and then prioritize your building block selections in a very rapid fashion. And you could send that directly to your chemistry team. And hopefully what that would do would enable you to really rapidly focus in on interesting chemical space. So, it's the end of the majority of my talk. We're here to help, whether you're looking to do this in-house with software, or you are looking to have someone else do this for you. A big part of my role is to do these kinds of activities for our clients, whether that's because you don't have the in-house expertise in computational chemistry, or whether you don't have the manpower, or perhaps this is an early stage project within your organization that you might be looking for some exploratory research without having to dedicate yourselves to in-house resources. So I'll have a expertise in virtual screening, library design, and these are all things that I can do for you, or which our sales team would be happy to talk to you about if you were interested in the software. So with that, I shall conclude, perhaps 5 minutes early, but I shall now open it up to questions from anyone, and perhaps say thanks for joining me today. So.
Okay, Alistair, can you actually hear me again?
I can, yes, Marcus.
Okay, excellent. So we have a bunch of questions, actually. It's great.
Oh, good.
Let me just walk through them one by one. One question was whether LeadIt is also capable of proposing replacement fragments. Perhaps you can comment on that.
Yeah, you can use a module of LeadIt called Recore to propose potential replacements for groups. I didn't talk about that at all because I think that was something else covered by you, Marcus, or someone else. And so, yes, you can. It's a very nice tool. And what you can do with the output from Recore is docking studies of the molecules it proposes, and perhaps rank order them by score as well as synthetic accessibility, because the fragments within the Recore database are based on crystal structures of real compounds, and so the geometries that are proposed and the bond connectivities are all very reasonable, and so it's a really nice tool.
Okie dokie. A next question is a very general question which came up. How could LeadIt potentially help optimizing log Ps?
Optimizing log Ps. Well, within LeadIT, perhaps easiest just to show you here. I'm still showing my screen now.
Yes, everything is good. We see your screen.
Good. So what I can do within LeadIt is I can modify my structure on the fly. So if I have a phenyl ring and I think, okay, I've got a problem with log Ps too high, what I can do is, I want to show this one, I can use the chemical modifier and say, okay, I want to make this less lipophilic. I can turn that ring into a pyridine. I can then save that and it will rescore it for me. So original score was minus 46, and just give it a minute to do the magic. No, so I picked the wrong position, obviously. So what you would do is then, okay, so that modification didn't work. We turn that one back into a carbon, and then you could work your way around your molecule, perhaps finding sites where that modification might be tolerated, and that would be one way of helping to reduce any log P issue you might have.
All right. So if that has not answered the question, then okay, here comes Tao. Yeah, Tao will unmute you, and then you can try to ask directly and communicate directly with Alistair. Just give me a second.
Okay.
Here we go.
Can you hear me?
Yes.
You can hear me?
Yes, we can.
So actually, this is what happened. I used LeadIT to improve my lead compound, and I got like negative 45 or something, really good score for it. But the thing is that I synthesized the compound. It took time for me to synthesize, like months. And then when I tested it, it was really like not soluble. And so if I did my best on LeadIT already, I mean height already, so what like, I don't think high can help me improve the log P unless I use some other tool to like vary the somehow vary the molecule so that like increase the log P value. Let me improve it.
Okay.
I mean, do you have any other alternative?
Well, you don't so much have the opportunity to calculate log P within lead IT. What you can do is use KNIME to do it, and there are a number of ways of calculating descriptors within KNIME. If you're looking to vary hydrophobicity within lead IT, is that something you'd be interested in doing? Accessing more buried pockets, or and I mean, it's hard for me to comment on specific cases, but generally, if you're looking to improve solubility, then doing the things like I mentioned, turning your phenyl rings into pyridines, and the standard medchem type of tricks like putting orthosubstituents in aromatic ring, distorting the crystal packing interactions, those kinds of things are things you can do and model within lead IT. So here, I could just, if we had an amide here, we've got an amide. If I wanted to methylate that amide, it's simply a question of changing that atom, and hopefully that will tell us, is that tolerated? And unfortunately, within lead IT, it doesn't give you a measure of log P.
Yeah, that's a problem because I didn't pay attention to log IP and spent months synthesize the molecule, and I thought that's the best I could do with the height already.
Yeah, so maybe I can comment on that from this position here. Actually, we are currently writing up technology which should be able to come up with estimations of log P. So as soon as you get output results, you should have an annotated log P value right next to it. This is currently, as I mentioned, in the writing, and thereafter we'll undergo testing, and then hopefully we can give this out to our fellow customers. For the time being, I think really there should be alternatives out there in the public for those of you who are interested in free software for these things. Tao, just in the interest of time, maybe we take further discussion on this very topic offline, and I proceed to the question.
Yeah, I mean, if you drop me an email, and I'll be happy to talk about your concerns and ways we could help you to move forwards and see what we can do.
Another question which came up was, could you recommend any patent search tools?
Yes. Unfortunately, most of them aren't very good. I tend to use, if I'm looking for a specific patent, you can go to the World Intellectual Property Organization, which is just WIPO.int. So that's one, and within this you can search for patents. It also contains the full text. Unfortunately, extracting useful chemical information from patents which have been machine translated is almost completely useless. If you want structures only, then you probably want to use a tool like SciFinder, but you have to pay SciFinder and the Chemical Abstract Society a lot of money to do that. In terms of what's available in the public domain, I mentioned Kemble, and they actually manually annotate a lot of data from literature, and so that's another resource which might lead you to patent molecules. But there's no, as far as I'm aware, there's no good free tool for accessing chemical information contained within patents. And it would be great if there was.
All right, thanks for this one. Then the general question arose, how does F-trees work in principle?
Oh dear. F-trees codes are what you might consider as being branches of a tree, and those branches are then compared to each other. And the technology, I mean, Sally is sitting next door, and she did most of this, I think. So my use is all as an end user, but it basically codes the chemical features of your structure as a string of branches, and then compares those branches. And so that's probably as much detail as I can give. It's, if someone wants to send me a message, I can surely put you in touch with our application expert, and they can fill in the blanks.
I mean, essentially, I can comment on this briefly. You're totally right there. It is essentially an alignment of trees, and the advantage there is, besides it being very fast, that we know why things are similar to each other. That's why we're able to actually color the trees in 2D, or color the molecules, telling the end user why something is considered similar to something else. So it's essentially, just as you said, it's a tree alignment which delivers this information there. What else do we have? I used leaded to generate analogs before. Every analog created has an ID starting with zinc. Wait, what does zinc mean? Maybe also
zinc is just probably the source that you downloaded the structure from.
Yeah, it depends maybe also a little bit on what tool actually was used. If you use Recore and then the results start with zinc, then this means that you essentially just use the sandbox fragment library, which comes with the software.
Yeah, sure.
And we certainly encourage everyone to go with larger fragment libraries, very diverse and large fragment libraries, and we'd be very happy to actually help you out there. There are various fragment indices that we can provide, but they are simply too big for just a simple download. So it needs a little bit of one-to-one communication if you would like to go with larger, diverse indices. So just drop us a note and we'd be there to help. Just going back. I think most of the questions unanswered in the chat have been answered. So did I forget anyone? Then please just retype a one-liner again into the chat box, and we can get back to answering. We still have 2-3 minutes.
I don't mind finishing 2 minutes early. If there are more questions, then it's I'll just say goodbye, and you can expect an email with your trial license, your 2-month trial license very soon. And if what I've talked about is in any way interesting to you, then all you have to do is send me a target PDB code or a small molecule, and I'll be very happy to run a small free pilot study for you with a confidentiality agreement in place if you want that, and try to convince you to use my service or our software. Just feel free to email me directly. You can find me on LinkedIn, and yeah, I'd be really pleased to hear from you. And so with that, I shall say good night and goodbye. So thanks for joining in.